With us today is Dr. Nisha Chhabria, and she is a movement disorder specialist, but she is actually involved in central nervous system gene therapy research through AskBio, and she is going to be providing our expert walkthrough and everything related to this gene therapy that they're doing at AskBio. So I am going to welcome you and hand this over to you, Dr. Chhabria.
Hi, everybody. As Anissa said, I am Dr. Nisha Chhabria. I am a movement disorder specialist, but I currently work in industry. I made the transition about 2 years ago. I work for a small gene therapy company called AskBio, where I'm their medical director for clinical development for their CNS gene therapy. Just give me a second while I share my screen, and we'll start the program. Hopefully, or so we think. Okay, here we go. So I told you who I was. So I do have to read this, sorry, by law. I am an employee of AskBio. I do not have any financial conflicts to disclose. I'm not promoting any service or product. And the references that are in here have been cited for publicly available information, copyrighted materials, and in figures and quotes. And I also have to read this to you again. I'm really sorry. This presentation contains forward-looking statements. Any forward-looking statement made by us in this presentation speaks only as of the date of this presentation and represents our estimates and assumptions only as of the date of this presentation. Except required by law, we assume no obligation to update these statements publicly, whether as a result of new information, future events, or otherwise after the date of this presentation. This presentation concerns product candidates that are or have been under clinical investigation and which have not yet been approved for marketing by the US FDA, European Medicines Agency, or other foreign regulatory authorities. The product candidates are currently limited by US federal law to investigational use, and no representations are made as their safety and or effectiveness for the purposes for which they are being investigated. All right, so let's get this started. So what we're going to start with is what gene therapy isn't. So that way you kind of get an idea of where we're kind of going to go with this. So the first thing is gene therapy is not CRISPR technology or gene editing. So I know a lot of you have heard of this, but that's not what we're doing. Also, we're not cloning. So we're not trying to make another copy of you or copy of anybody else. So no dollies in this case. We're also not looking to make designer babies. We're not looking to try to get you that perfect child that you're looking for, though I do kind of like the fact that the cartoonist is 20% off. And also, we are not stem cells. So I know a lot of people have heard about stem cells. That is not what we do. We do something completely different. And so we will talk about that next. So what is gene therapy? What is it that we are doing? So let's start with what are genes? What are genes exactly? Well, genes are part of our DNA. They're basically what carry our genetic information. And we each have 2 of them, and we get 1 from each parent. And according to the Human Genome Project, we believe that we have about 20,000 to 25,000 genes. And about 99% of those genes are common between all of us. Only about 1% of those genes are what differentiate us from the other people around us. So that's what makes our uniqueness. The important thing about genes as well is they're really what become the proteins in our that we're made up of, and what do become medications in the future. So let's do a little bit gene therapy 101. So like I said, genes are basically our DNA. They're our blueprint. So they're basically the instructions to our cells to kind of
Okay, I think we froze a bit. I'm going to give her a second to see if it self-corrects. Sometimes it will. There you are. We had you frozen for a second.
Sorry, my Wi-Fi has been acting up all day. I apologize, you guys. Where did you lose me, and where can I go back to?
You were literally just talking the first bullet.
On this one, okay.
Yeah.
Okay, so going back, so genes are basically our blueprints, right? So they are what we need to be able to give instructions to our cells so that they can grow and be healthy. So when we have genes implemented into cells, these cells then read the genes, or as we're calling them, instructions on how to make new proteins. These proteins are then used to signal the health of a cell and then also how to keep them alive. We also are using gene therapy in this regard to help make those proteins, because those proteins are then acting as a medication to treat disease processes, or in this case, Parkinson's disease. So here at AskBio, what we do is we use a capsid called AAV, and that is an adeno-associated virus. And we are known for this virus, and actually many different companies have used our virus. And this virus is what we use to deliver the information to the cells to make the new protein. So that information, as we've been talking about, is the gene. So basically, as you see here, we have the capsid, and then inside it carries the gene. So basically, we're trying to find a way to deliver this gene into our cells. And so one of the important things about that is that this virus that we're using has been modified so that it doesn't make people sick. So basically, I'm about to explain how this really does work in a different variation of ways. So kind of through imagery, but also through some videos as well. So why do we use the AAV2 in the brain? Well, like I said, this is something that's been used for many different diseases, and it's been determined to be pretty safe. We know that it's effective and it's an easy way to get genes into the brain. Like I said, it's safe, and we know it doesn't cause infection, as it's been modified not to cause infection, and it has a lasting effect. So let's talk about how this virus is delivered into the brain. So basically, let's talk about the gene. So our gene is our letter. Let's talk about it as if we were going to go to the mailbox and deliver a mail or a letter to somebody else. So the genetic material, or the gene, is our letter. The capsid, the AAV, is our envelope. We put the letter into the envelope, we package it up. We then give it to the delivery truck, which can be delivered in different ways. In regards to the brain, we deliver it surgically. Sometimes it's also done by an infusion or through an injection if it's being done for other parts of the body. And then the delivery truck delivers this to the cells, and then the cells then read the protein or the gene that's been delivered to them, or the letter that's been delivered to them. And then they take that gene and they make it into different proteins. And so I'm just going to kind of guide you through some videos here. So as you can see, that's our gene, and that's our vector. And now that gene is going to go into the vector, and that vector carrying the gene is then going to get incorporated into the cell. And then that cell reads that protein, as you can see, or that gene, I'm sorry. And then that spreads it out as a protein throughout the entire nervous system. And in regards to what we're working on, we work on something called GDNF, or glial cell-derived neurotrophic factor. GDNF is a protein, but it's also a gene. So what we do is we take the GDNF gene, we incorporate that into the vector, and that vector is then, like I said, delivered into the cell using a surgical technique. And then that cell reads the gene of GDNF and breaks it down into GDNF proteins, or small amounts of GDNF protein, so that those cells around where it's gone into can regrow. So basically what it's doing is it's taking the dying dopamine cells and trying to bring them back to life. So the ones that are already dead, we can't really do very much for, but for the ones that are on the verge of death or have the potential to regrow, we're basically trying to get those cells to come back to life and produce dopamine that the body needs, or especially in a Parkinson's patient. This isn't going to get us back the 100% amount of dopamine that we would normally need, specifically because those cells, there are dead cells that can't be revived, but it's trying to take the other cells and bring them back to life so that you can at least get that 30 to 40% of dopamine that needs to be produced. So why are we using this gene for gene therapy? So like I stated, this gene has been shown to show resprouting abilities. And we know that that's something that can be very beneficial for Parkinson's patients. And at the moment, we really don't have anything more than just medications. And these medicines just kind of keep us in an even keel state. But over time, they lose their efficacy, and we need different medications, or we need to take medicine closer together. And so this may be something to help reduce the amount of medication that a patient will need to take in their lifetime. This is not used specifically for a genetic type of disease. So I know with Parkinson's, we can think about LRRK2 or GBA. This is not targeting those genetic forms of Parkinson's. This is targeting any type of Parkinson's disease, because this is gene therapy and this is not specific to a genetic type of Parkinson's. And the great thing about this is it's a one and done therapy. So you do it one time and you won't have to repeat it again. So what do we know about GDNF and Parkinson's? So I've kind of touched on the fact that we know that there is this loss of GDNF in these patients. And as we deliver this, we're trying to get the restoration or neuronal like health to be able to restore those sick and dying cells. So the goal then there is, this is not a cure, but to maybe try and slow down the progression of the disease. So that's what we're trying to do with this therapy. So how do we get it into the brain? So this looks very daunting, I know, but essentially what we do is we use MRI guidance. So the patient will lay prone on their stomach for several hours under general anesthesia. And we use a bilateral parieto-occipital approach, so coming in from the back of the brain at the same time, to then go into the brain and deliver the medication into the putamen at the same time. And so I'm going to show you a little bit about that in a few minutes. But basically, like I've said, this has actually been done over in the MRI machine, and this has been done safely over the last 15 years. So this is a simplified version of how we do this, but basically we take a very hollow tube or a catheter, as you can see here, and then we infuse as we go, and we can try to infuse the therapy into the putamen. And like I said, it's done one time and it's never done again. And so you can see that we use gadolinium, which is the white stuff, and this is the medication. So this is the putamenal area here. And this is where we have our two cannulas, and they push forward and infuse, push forward and infuse, push forward and infuse. And we do this in real time, and we can do this, and this takes about 4 to 6 hours to complete. And this is going to show you exactly how we do it. And there are actually two cannulas in the video, but this one's the only one that's in the field of plane of field. This one is not, but you can see that this is actually an infuse as you go, as I keep saying, and it's very cool because this is done. This is a speeded up version. Obviously, like I said, this takes 4 to 6 hours, but it's a cool infusion that's done at the same time. So it's really a much quicker procedure than having to do them separately. And our goal for all of our studies has been to get greater than 50% coverage of the putamen. And I will be going over some of our data, so you'll actually get to see some of the information. So a typical surgery day, important thing to know is on the day, you're definitely not going to have any hardware like you would with DBS. There's nothing to have to worry about maintaining over time. After the surgery is over, you stay in the hospital about 1 to 2 days, depending on how the surgery center likes to do their things. And then because this is done in research, you are seen and followed up every 3 months. For at least the first 18 months, and then after that, you're seen every year in the study and depending on which study, we're talking about anywhere from 3 to 5 years. All right, so let me talk to you a little bit about, well, before I move on, does anyone have any questions? Is there anything in the chat that we wanted to discuss about the previous?
So what we've come up with questions, first is there was a question, AB-1005, is your firm's phase 2 testing correct? Is that what this is?
Yeah, well, I'm not talking about specifically the phase 2, but AB-1005 is the actual investigational product. So it's the product that we've used across all of our clinical trials for Parkinson's and multiple system atrophy.
Okay. And Another question, is this just for Parkinson's or Parkinsonism? How do you know if someone has idiopathic Parkinson's when sometimes it takes a while to designate an atypical?
Well, so we are doing this study for both Parkinson's and multiple systems atrophy. So we actually are using it for two different Parkinsonian-like syndromes. Idiopathic Parkinson's, well, we call it idiopathic specifically because idiopathic means we don't know what's causing it. So in some clinics, they'll actually test you for genetic causes of Parkinson's. I mean, not every clinic will do this. Or if you've ever done 23andMe, it does test for the two most common forms of genetic forms of Parkinson's, LRRK2 and GBA. But the best way to get that diagnosis of is this Parkinson's or Parkinson's like Parkinson's plus is to be seen by a movement disorder specialist. They're the ones who have the most expertise in movement disorders, specifically Parkinson's, MSA, Lewy body. But there are certain things that kind of can give you a little bit of a distinction between like a Parkinson's versus an MSA or a Lewy body. For multiple systems atrophy, a lot of times patients will have early onset of feeling lightheaded going from sitting to standing, where their blood pressures are going to drop dramatically when they change positions, or they're having earlier cases of urinary incontinence, retention, frequency, waking up frequently overnight to use the bathroom, excessive sweating, erectile dysfunction, things that are called autonomic dysfunction. Those tend to come sooner than they would in an idiopathic Parkinson's patient. So sometimes you can use that to kind of help determine if this is MSA versus Parkinson's disease. Levodopa responsiveness actually is another thing that a lot of doctors will use to determine if this is Parkinson's or if this is something else. A lot of times an MSA patient will respond at the beginning, but after a couple of years, maybe they'll have more of the orthostatic feeling, or they just notice that it really doesn't make that much of a difference. So that kind of helps guide the physician as well. For Lewy body dementia, there's generally a sense of cognitive problems, so trouble with your memory. A lot of times patients will say, I've had problems with my memory, and then they kind of had memory testing. It showed that they had a little bit of memory problems, and then they started developing the symptoms of Parkinson's with tremor, slowness, stiffness, trouble walking. A lot of times just giving them levodopa with Lewy body, they hallucinate almost immediately or within a couple of months of being on the medication. That's another way to help with making these diagnoses. So there are certain things that we can do, but generally, yes, at the beginning, a lot of people are diagnosed as Parkinson's until subtle signs kind of come out for us to make that diagnosis. And it can take a couple of years to get to that point.
And does having DBS preclude somebody from participation in gene therapy?
Yes. Unfortunately, this is something that you will not be able to participate in if you've had deep brain stimulation. Now, if you do participate in this study and in the future, if you decide you do need to have DBS, this study or any of these studies will not prevent you from being able to undergo DBS in the future if that's needed. Obviously, our goal is to make it so you don't need that, but we have at the moment only 5 years worth of data that's showing stability, but we can't really say for sure what the future would hold after that at this point.
I do have other questions, but I'm going to hold them to see that you're not going to go ahead and answer them, and then I will come back to you with those.
Sounds good.
Mhm.
All right, so I'm going to kind of talk to you a little bit about what will be out into the public hopefully soon. We did present this at the American Academy of Neurology in April, but this is talking about our phase 1B study that has closed. We had a total of 11 patients in that study. So basically the way that study worked out is we had what was called a mild cohort and a moderate cohort of Parkinson's patients. Like I said, we had 11 patients. The average age was about the same between the two cohorts. For the mild cohort, they had a diagnosis duration of about 2 years, and for the moderate, it's about 8. For the MDS UPDRS Part 3 off scores, for the mild, it was started around 20, and for the moderate, around 40. And for the UPDRS on score, they definitely had robust responsiveness to levodopa of almost or greater than 50%. And if you kind of look at what the levodopa dosing is for these patients, it's on average of what you would see for the type of cohort that you're looking at, with the mild at about 500 milligrams and the moderate at about 1100. So this is what I was talking about with regards to trying to get the coverage of the putamen. Our goal was to get to about 50%. We actually went above and beyond that to 63%. And this was about the same in both the mild and moderate cohort. And we're just showing you the video again so you can kind of see how we do the infuse as you go. When we look at the side effects from this medication, it's kind of like, or not the medication, I'm sorry. When we look at the side effects that we saw in the study, it's what you expect for somebody who's undergoing an operative, a surgical procedure. So before they had their surgery or during the surgery period, we did see that they had, the patients experienced headaches, muscle aches, fatigue. Those are the things you would expect for someone who has had to lay flat for several hours in an MRI machine, and also had holes drilled into the back of their head. And then the tremor we also saw because they weren't taking their medication for several hours, and then by the time they were able to actually get their medication on board, it did take some time for the medicines to kick in. And then in the post-operative state, we did see that they had the same sort of kind of muscle aches and fatigue. And then this was done during COVID. So it's expected to see COVID-related, side effects. So this is kind of the cool thing that that's one of the reasons why we're kind of chasing the moderate cohort for our other study, is what we see here in the mild is that there's kind of stabilization across the board in their assessments. The reason you see this one outlier over here is that patient turned out to have a tyrosine hydroxylase deficiency. We did not know about this before the study was conducted. It was found after the study, the patient had already been enrolled. But if we take this person out, we'd actually see more stable lines across the board. But one of the reasons we're chasing after this cohort, the moderate cohort, is because if you see, we see that they started at a 40 with their off, and now they're down at about a 20 with their on, which is about a 50% improvement over 18 months after having had the surgery. And same across the board with the other two, the on and the part 2. So because we've been seeing this dramatic improvement and then some stabilization between month 12 and month 18, we decided that this is a group of people that we would definitely like to pursue in the future. One of the other things we had our subjects look at is their PD Hauser diaries. I'm sure all of you have completed this or know what this is. It's where they give you a table and it asks you if you can tell me if you're off, if you're on, if you're having dyskinesia, if they're troublesome. And then also, of course, while you're sleeping. So it tells us how long you've slept and what you've been experiencing. And so what we see here is that we're looking specifically at the moderate. They had about 4.5 hours of off time. And by month 18, they were at about 2 hours of off time. So there was definitely a dramatic improvement in the off time compared to baseline. And there was also an improvement in their on time, as you can see as well. We went from 10 hours of on time to almost 14 hours of on time with very minimal troublesome dyskinesia. So this is a big deal. And some of these patients were able to actually reduce their levodopa dose as well. So what does this mean? Like, why did we do this study, and what does this mean for the future? Well, one thing you guys do need to know is this study was done as an open label study, meaning that every single patient in the study did receive the medication. And what we know with Parkinson's studies is that there is a very large placebo effect in Parkinson's studies, specifically because we know that dopamine controls Parkinson's. And even if you get the idea of thinking that you got the medication, there's the potential of thinking that your brain kind of makes that dopamine in the anticipation of hoping that you got medication. And so, we do know that everybody got the medicine, but there's still the chance that there is still a placebo effect in this case, because they all knew they got the medication. So it's possible to say, well, because we know we got this, then our brains are saying, oh, we're going to make more dopamine because we got something that's supposed to make us better. So we definitely need more studies to see if we can do this in a placebo-controlled way, so that way we can determine if there really truly is an effect, or if it really is truly just a gigantic placebo effect. And then, we did see, as I showed you in our mild group, there seemed to be some stabilization over the last 18 months. And then with our moderate group, we did see some improvement over the last 18 months. And now these patients who did participate in this trial are now about 3 years into the study, and we are seeing stabilization and improvement, and that's a big deal. But now what we're trying to do, and we're hoping to get underway, is a blinded study so that we can truly prove that this is a drug that is going to change people's lives. So what do you, let me stop here and see if I answered any of those questions that you had.
Okay, so one question was, what is different with the current study from previous using GDNF?
Good question. So delivery. So the GDNF, I mean, for those who've been in medicine for a long time or practiced medicine for a long time, you've probably heard of GDNF. And unfortunately, every GDNF trial has failed. And it's because of the way the GDNF has been delivered. The most recent GDNF study was done in the UK, and it was a protein infusion study. And that study actually had to get shut down because it ended up causing more harm than good. And we found that it actually did not have any benefit whatsoever. Now, before the phase 1B for AAV-GDNF was done, there was a phase 1 done at the NIH. And we actually learned a lot from that study because, again, it had to do with delivery. So we were using the AAV-GDNF technology, but it was delivered in a very different way. It was delivered frontally instead of from the back, and they weren't able to get as much coverage of the putamen. So what we're finding is that delivery using the AAV-GDNF techniques, so using a viral vector, and then also using a different approach going through the back of the brain, helps get more coverage to the putamen, which then gets you more of the GDNF to the brain to be able to get it to do what it's supposed to do.
This person wants to know what actual gene is used that leads to increased GDNF. How does it lead to sustained preservation of dopaminergic neurons if it is not incorporated into the genome?
So that is, well, so it is GDNF. So the gene is the GDNF gene, and it is being incorporated into the genome. That's what's happening when it's being delivered through the AAV2 vector. So the vector is the delivery system, or as we said, the envelope that gets it into the cell. That breaks down, and then the cell will read the gene that's being implanted, which is the GDNF gene, which then sends out the GDNF protein to help revive those dying dopamine cells.
So we just had a question going back to, I guess, who is appropriate. Are people who have no distinct on-off times candidates for this?
No. In the phase 1B, they would have been because there was a milder cohort. So there was a different set of criteria with regards to that. But with regards to this, our study, we are looking specifically for patients with greater than 2.5 hours of off time.
So I know your next section is how to participate in clinical trials. So I'm going to hold some of these questions related to the clinical trials until after, just to make sure you're not going to answer them. So we have those, though. I just wanted to let you know.
Okay. I probably won't, but we'll go.
Okay. Well, that's all right. We'll come back to them.
Sounds good. Okay. So, well, obviously the first thing to do is talk to your neurologist if they know about them. And a lot of movement disorder specialists, if you guys are seeing a movement specialist, do research. So they do know what research studies are being done, and it's always good to talk to them about it first. And then the next thing you can do is use clinicaltrials.gov. And they are an excellent database for looking for any type of research study. You can just type in Parkinson's disease and it will just pop up a whole bunch of different studies for that, or you can type in a specific type of trial that you're looking for with either the name of the company that's running it or like a specific therapeutic area. So meaning like motor symptoms or non-motor symptoms, or just like type in urinary continence, Parkinson's or something like that and see if there's some trial for that. And so basically this is where you would do that. You can put in the condition. Here in the other terms, you can, if you know the number, you can put that in. You can put in the name of the company. You can even specify which country you would like to do it in. And then also the Fox Trial Finder is another excellent way to be able to find similar studies as well. Same kind of concept, you just type in the name of the trial that you want and then it will pop up for you. So just real quickly about who we are. So AskBio is the leader in next generation AAV2 gene therapy, and we're a pioneer in almost every element of design, application, and manufacturing of AAV therapeutics and technology. We were founded in 2001 based on 40 plus years of research by Drew Samolsky. Interestingly enough, actually, I was at ATMRD. I actually spoke to one of my old attendings, and he actually worked with Jude on AAV technology, which was an interesting story to hear about. That was kind of cool. And our pipeline at AskBio covers a range of genetic disease targets, not just for neurological conditions, but for muscular, respiratory, and heart. We have over 500 employees, and in the US, we're based out of RTP in North Carolina, Columbus, and Philadelphia. And in Europe, we're based out of Edinburgh, San Sebastian, and Paris. We're a comprehensive novel technology platform with 800 plus patients from discovery through delivery. And like I said earlier, our technology has actually been acquired or licensed by different companies such as Pfizer, Takeda, and Novartis. So if you're interested in any of the clinical trials that we may be running, and it doesn't just have to be for CNS, we have, like I said, clinical trials in neuromuscular disorders, in congestive heart failure. We have a program, it's called Ask First, and there are patient engagement, patient advocacy, and they are happy to inform you about anything that's going on with AskBio at this time, and they are a great resource for anything regarding all of our clinical trials. And the email for that is right at the bottom. It's askfirst@askbio.com. And so we would encourage you to consider participating in a clinical trial, because without you, we can't make the lives of other Parkinson's patients better. And we also want to say thank you to anyone and everyone who's participated in any of our clinical trials in the past. Okay, that's it.
Okay, so I'll go back to some of these questions. So we had a question about whether you are still recruiting patients for the study. And related to that, is it available only in Kansas?
So yes, we are actively recruiting for our phase 2 Parkinson's study. Clinicaltrials.gov should be updated to show that Parkinson's Disease and Therapeutic Center of Southwest Florida in Port Charlotte is also open as well. So at the moment, yes, those are the only 2 sites that are recruiting, but we will have more sites up and running at the end of July, and we have multiple sites that will be starting in the month of August. Our sites eventually will be in California, Las Vegas, Kansas, Texas, California, Las Vegas, Kansas, Texas, Wisconsin, Michigan, Florida, Pennsylvania, Boston. I'm trying to think what else. And North Carolina.
Okay, so you have quite a few locations. It is all over the place.
It is.
There was a question, is there other countries that will be participating in this, such as the UK?
Yes, so the UK, Germany, and Poland will hopefully be participating in these studies. We're actually in the process of getting regulatory acceptance from all of those countries as we speak.
So this question is, phase 1B is early testing of this therapy. If everything goes well, when would this therapy be available for actual distribution for PD patients? So maybe a little like background on how that whole process actually works from development to market.
Exactly. So we are in our phase, we did our phase 1B, and we are starting our phase 2. Ideally, we will have the entire study recruited in the next 3 years, which would get us to about 2026, 2027. And then obviously we would have to unblind. This is a blinded study, so we will have to unblind that study after the last participant hits the 18-month visit, and then determine if this is a safe and efficacious drug and something that we could even move on to with a phase 3 study. If that's the case, then we will actually be able to consider moving on to a phase 3. And once the phase 3 has been determined to be safe and efficacious, we can then apply for FDA approval. So ideally, in a perfect world, we're hoping for 2032.
Gives us at least some idea. So I know that for the treatment or for the actual trials, DBS is not even possible, but can you foresee this ever being available when it hits market for somebody who's had DBS, or is that something that's just because of the type of surgical
Yeah,
involvement of DBS, it wouldn't work?
I don't know the answer to that. I would believe you would probably have to turn the DBS off in order to actually get the benefit from this therapy. And given that we are not trialing it in patients who have DBS, on the label, it will not allow patients with DBS to undergo this procedure. So I can only speculate, but I can't really say if this is going to be something we would be able to use in those patients.
So I know you mentioned that you're doing a variety of different types of trials at AskBio. Are you doing any trials in other movement disorders other than Parkinson's?
Multiple systems atrophy.
Okay.
Mm-hmm.
Awesome.
So that one's actively recruiting as a phase 1/2.
It's exciting. I've caught up with the questions. I think I will open it up again and encourage you, if you have any burning questions about this, now is your chance to ask them. This has been fascinating, and it's exciting how many different locations you're actually going to have available for this trial.
Yeah, we're excited, and hopefully they'll all be up and running by the end of this year. But by the end of August, we'll definitely have a good portion of our sites up and running, which is a big deal. The important thing is we have a concierge service. So even if it's not in your state and you are interested in doing this study, this concierge service will get you to those sites that are participating in our clinical trial. And so you would be able to have them get you where you need to go. And they're going to be active in helping you with this study to begin with, because the surgical sites are much fewer than the neurological sites. So you will have to travel for the surgical procedure. And then we're also doing central imaging for PET scans, and you have to travel for that as well. So the concierge service will be your right-hand person to get you wherever you need to go and get you housing and meals, all of that kind of stuff. So
Wonderful. I had a question of where in California will you be?
as of right now, it will be Loma Linda and UCSF.
So I'm going to add to this question that just came in from David. They want to know, as I think you've mentioned a few of them, but can you go over all of the inclusion criteria? Because I'm getting a question about age restrictions. So I know age is always a consideration for inclusion or exclusion. So can you review those?
Yeah, the age limit is 45 to 75. You have to have been diagnosed within 4 to 10 years. Anyone before 4 years would be excluded, and after 10 years would be excluded. Your MDS-UPDRS off score, so the assessment that your doctors do on you, that has to be between the ranges of 33 to 60. The modified Hoehn-Yahr score, which is what we determine your staging, has to be between 2 and 3 for in the off state as well. You have to have greater than 2.5 hours of off time. You need to be on at least 300 milligrams of levodopa, and it has to be stable for at least 4 weeks prior to entering the study. And you have to have levodopa responsiveness, meaning you do have to know that there is an improvement when you do take your levodopa versus when you don't take your levodopa.
And because many people probably don't necessarily know their UPDRS score off. Some people might, but not everybody does. I'm assuming this is something that you would be discussing, like potentially with your treating healthcare provider and movement disorder specialist, hey, I heard about this. Looks like there is a location relatively close. I'm interested. Do I meet the criteria? Would it start there, or do they just reach out to the study coordinator to find
I,
out if they fit? How does that process work? If they've identified, yes, this is here, I'm interested in it.
Yeah, definitely start with your neurologist. I mean, they actually, if they are doing the UPDRS, they can give you a general idea of like a number. And they can also kind of guide you if they feel like you have a responsiveness to your medication. And we do have recruitment materials like that the sites will have to give to outside physicians around them and that kind of thing. And also, really, to be honest, clinicaltrials.gov has a lot of what we talked about. It's on that website, so you can get a good idea as to whether or not you are a good candidate for this. But yeah, also, reach out to the sites, because once they're open, you can easily go in and figure out where you can go and reach them through using clinicaltrials.gov, or go through Ask First. We're happy to help you in any way we can as well. They may even be able to give you some of the recruitment material too. Basically, that's the best way to start. And then, the site can also send you the informed consent so you can get an idea. It's a very long informed consent, guys. It's about 60 pages. So it's not something I would say just come to the office and read it. Like, you should be given it well in advance, specifically so that you can actually read it, review it, figure out if this is something you can actually achieve and do, and then go to the site to actually sit with them and review it with them again and sign it. So it's a hefty consent form for sure.
Like, have to be committed.
This is a big commitment. Unfortunately, this is a very, very extensive procedure. Yeah.
Absolutely. Well, I appreciate you giving all of this information. It's exciting to see where we're headed. I know it's early. I know it might be a little disappointed to think it could be 2032 before we see something to market, but honestly, it's exciting that we're at the place that we are enrolling in a phase 2, that there's going to be so many places that people can engage in that trial, and that we have the opportunity to actually hear from you about this early on and be able to ask the questions. So with that, I'll ask one more time, if there's any unasked questions out there, please feel free to drop those in the chat. And give you that last opportunity, because we still have time. And I agree, it is cool to see what science is able to do and the new and innovative treatment possibilities, as Rebecca said. So, and we appreciate people like you, Dr. Chhabria, that's out there doing these trials and educating us about them.
So interestingly enough, the phase 1 is the reason that I actually wanted to work with this company. So that in itself just kind of made me excited. So
It is really exciting. Well, I think we must have come to the point of everyone's questions have been answered or addressed through your very comprehensive, your slides and everything that you've explained. So I want to thank you so much for that. This has been fascinating. I've learned a lot. I'm sure everyone else has too. It's very exciting. So guess I'm going to invite everyone to turn on their cameras if they're not on, because I don't know that you've actually presented for us before, but we have a tradition at PMD Alliance. We love our speakers. And so we love to give them some gratitude from across the miles. And so if you have your cameras on, give Dr. Chhabria the wave of gratitude for this. Thank you.
thanks, guys. It's very sweet. There is a question in here,
Yes,
though.
I was just going to say, I did see that coming in. You want to go ahead and answer that?
So the funding for the study is AskBio. So we're the sponsor for the study. And the requirements, will they change once made available in 8 years? That's a really good question. Probably a little bit. Obviously we do have to stick with like, yeah, just a little bit. We're gonna have to stick with like certain aspects of it, obviously like a moderate stage Parkinson's patient, modified Hoehn-Yahr of 2 to 3. Would doctors consider using it in older patients? Probably. I would say that that's, I'm sure with DBS when they did their studies 20 years ago, they had restrictions on their ages. And I mean, we've done DBS in patients who are 90. So I'm sure that would definitely change in that regard as well. But I think we would be looking to start using the procedure in patients who are more moderate versus the mild group. Would we eventually try to get to that? Obviously, yes, that is our goal because we do have data with regards to a mild group. But labels are a way things get paid to start. And so we would probably have to start with on label first, going off label would be later.
All right. Well, thank you so much. And we hope you have a great day. Hope everyone else has a great day. See you soon.